Hello and welcome to the third video on the human biology course. In this video, we will analyze the mechanisms through which cystic fibrosis affects the human body. We will also explain certain concepts related to lung function, gas exchange, and cell biology. Let's start by taking a look at the anatomy of the lungs. The lungs are part of the respiratory system, which starts at your nose and mouth and follows down through the trachea, which then splits into two bronchi. The left bronchus is part of the left lung, and the right bronchus is part of the right lung. The bronchi then become narrower and are called bronchioles. At the end of the bronchioles there are alveoli, which is where the gas exchange occurs, allowing us to breathe. There are around 300 million alveoli in our lungs. I bet you're wondering how on earth we've got space for all that in our chest. But we do, and that is because each alveoli is extremely small, around 250 micrometers in diameter. That is 0.25 millimeters, so they really are very small. The reason for this is because in order for gas to be exchanged, we require a large surface area. The more of these alveoli we have, the more surface there is in our lungs for this exchange to occur. If you think about a small box and you put 10 balls inside of it, if you were to stretch and unite the material of those balls, it would be fairly big. However, if instead you filled it with 1,000 smaller balls, you would be able to not only fill more space in the box, but if you stretched and united the material, it would be much, much bigger. That is the principle behind the anatomy of our lungs. An increased surface area means there are a lot of blood vessels present. These vessels become capillaries in the alveoli. When you breathe in, you are transporting a mix of gases from the air into your lungs. In there, the air travels through the lungs until it reaches the alveoli, where the oxygen in the air enters your bloodstream through the capillaries. The blood then transports the oxygen and spreads it throughout your body. When oxygen is spread through the body, there is an exchange. Oxygen enters the cells from the bloodstream, and carbon dioxide leaves the cells into the bloodstream. So when you exhale, the blood flows into the capillaries in the alveoli, and the carbon dioxide leaves your body as you breathe out. When you breathe in air, it can contain dust, germs, and other particles which we do not want to enter our body. That is why our airways are coated with a layer of mucus, which traps these particles. Special structures in the airways called cilia, which look like hair, then move the mucus which contains those particles out of the airways, keeping our lungs healthy. Proteins, which we have talked about in previous videos, have a multitude of functions in our bodies. Some proteins are attached to the cell walls, facilitating the movement of ions and molecules into or out of those cells. In the cell walls of the alveoli, there is a type of protein called the CFTR protein, which is short for Cystic Fibrosis Transmembrane Conductance Regulator protein. I know, it's a mouthful. Which facilitates the movement of chloride, present in salt. When this ion enters the cells, it attracts water to the area. This water is one of the components of that mucus that covers the airways to catch unwanted particles and germs. It basically helps maintain the mucus at the perfect thickness to trap those particles. However, in cystic fibrosis, that protein that transports the chloride ions into the lung cells is mutated, which means that it either does not work well, that the protein is produced in insufficient amounts, or that it isn't produced at all. This then leads to chloride becoming trapped inside the lung cells, which in turn does not attract water to the area, making the mucus very thick and sticky, which then means those hair-like cilia are unable to move the mucus away from the airways. As you can imagine, this then leads to difficulty breathing, to the accumulation of particles and germs in the airways, which can give rise to a number of infections and diseases, and inflammation of the airways. A similar thing occurs in the digestive system, which we briefly mentioned in the last video. Similarly to the lungs, the passageway that connects the pancreas to the small intestine becomes blocked in a person with cystic fibrosis. The same process occurs there, in which chloride is transported into the cells through CFTR proteins, which then attracts water, making the mucus the right consistency and thickness. In cystic fibrosis patients, these proteins are defective, trapping the chloride ions inside the cells. Therefore, the mucus becomes thick and sticky. The lack of digestive enzymes being transported into the intestines due to this blockage then means that food remains undigested, giving rise to pain, cramps, gas, and overall an unhealthy gut. Now let's have a look at cell biology and how cell exchanges occur. Human cells are eukaryotic. This means that our DNA is enclosed in a nucleus, and the cell organelles as well as the nucleus are enclosed by a membrane. Let's look at a diagram of a eukaryotic cell. As you can see, there are several organelles within a eukaryotic cell. The main organelles we are going to focus on today are the Golgi Apparatus, vesicles, ribosomes, the endoplasmic reticulum, both smooth and rough, and the mitochondria. As you may know by now, proteins are made from specific DNA sequences. The DNA is a double helix constituted by 4 bases: adenine, guanine, thymine, and cytosine. Adenine pairs with thymine, and cytosine pairs with guanine. The order in which these bases appear on the DNA chain is what gives rise to the countless proteins which we produce. In order for these proteins to be produced, the DNA first needs to be analyzed and decoded in a process called transcription. This transcription is then put in place, and the proteins are then formed in a process called translation. We will touch on this in a later video. Ribosomes are involved in the translation phase, where they decode a strand of messenger RNA into a protein. Ribosomes can be found either in the cytoplasm, or they can be found attached to the rough endoplasmic reticulum. The rough endoplasmic reticulum, or RER, is another organelle involved in the synthesis of proteins and also in the synthesis of lipids. It gets its name because of the ribosomes which are attached to it, giving it a rough appearance. In this organelle, proteins undergo the final stage of their synthesis, where they gain their specific structures and functions, and this is where their destination is determined. They either remain in the endoplasmic reticulum to form part of its membrane, or they are transported into another organelle through vesicles. Vesicles are the taxi drivers of the cells, as they transport proteins and other molecules into and out of the cells, as well as within the cells. Another important process also occurs in the endoplasmic reticulum, that of quality control. This means that any proteins which are defective should be detected by this organelle and consequently rejected and recycled. The smooth endoplasmic reticulum is mostly involved in the synthesis and metabolism of lipids. Finally, the Golgi apparatus is basically the logistics department of the cell. It determines where the proteins will go after, and minor changes are made to allow proteins and lipids to become fully functional. Now let's correlate the functions of these organelles with cystic fibrosis. As mentioned before, this disease arises from a genetic mutation. There are actually several mutations which lead to cystic fibrosis, but we are focusing on the most common one. This means that during the transcription and translation processes, the CFTR protein which is synthesized is slightly different than what it should be. This mutated protein is then transported into the rough endoplasmic reticulum, and it is there that the problems begin. During the quality control phase, this organelle detects the mutation in the protein and therefore it doesn't allow it to leave, which then leads to a shortage of CFTR proteins, giving rise to cystic fibrosis. The protein never reaches the Golgi apparatus and is recycled, and the whole process repeats over and over again. Finally, let's talk about some concepts you should know about regarding the means of transportation into and out of the cells. The membrane that surrounds cells is called a plasma membrane. It is composed of a lipid bilayer and proteins. The lipids which are present in the membrane are mostly phospholipids, and they have a hydrophobic end, so basically this part of the molecule is afraid of water, and the hydrophilic end. This is the water-loving end. This is why it is a bilayer. As you can see from the picture, the hydrophobic ends face each other and the hydrophilic ends face the outside and the inside of the cells. The proteins in the membrane can either cross it from one end to the other, in which case they are carrier proteins, as they can help carry molecules into or out of the cell. Proteins can also be attached to one of the ends of the membrane. Now let's introduce three concepts: facilitated diffusion, active transport, and osmosis. Facilitated diffusion, like the name says, is the movement of molecules with some aid, hence facilitated. This aid is either through a channel protein, which crosses the membrane, or a carrier protein. This movement occurs according to an electrochemical gradient, from high to low. Let's think of it like two football teams which are separated by a door. One of the teams has 15 players and the other team has 7 players. Now, we know that in football each team has 11 players. Therefore, 4 of the players in the big team pass through that door and join the small team, evening the numbers out. This is the principle of facilitated diffusion. Molecules and ions go from a high concentration area to a low concentration area with the help of proteins, and no energy is required in this process. Active transport is the opposite. It requires energy as it goes against the gradient from a low concentration area into a high concentration area through proteins across the lipid bilayer. The energy required to undergo active transport is called ATP, and it is produced in the organelle called mitochondrion, or mitochondria in plural. Osmosis is the same as facilitated diffusion, but without the use of proteins. In this process, solvents such as water pass through the lipid membrane according to an electrochemical gradient, from high to low concentration. As with diffusion, no energy is required for this process. And that is it for today. I hope you have enjoyed this video. You can check out the whole course on our Patreon. See you next time!